This video is going to take us through Lab 9, which covers the Introduction to Genetics chapter in your lab manual. To start us out, we're just going to go over some definitions and some important people. Well, an important person, I suppose. Genetics is the study of how traits are going to get passed down from one generation to the next. Mendel is the person who helped us really understand how traits got passed down from one generation to the next. He was an Austrian monk. He lived at the same time that Darwin did, but he published in different journals, and unfortunately neither of the two, well, Darwin never read Mendel's work, and so a lot of questions could have been answered had these two actually met each other. That would have been really fascinating, but they didn't. And so they really didn't get to compile their data. The other issue is Mendel published all of his results in a fairly obscure journal, and it was hidden for decades before somebody rediscovered it. And it really helped everybody understand how traits did get passed down from one generation to the next. Genes, we kind of talked about these in the last chapter when we were talking about protein synthesis prior to the first lab exam. Remember that genes are specific stretches of DNA or part of a chromosome, and they code for a particular product. In the protein synthesis chapter, we of course talked about genes coding for proteins, but they can code for other things as well. Next up, you have a couple of terms, genotype and phenotype, and I'm just calling them the types. Genotype is the actual genetic makeup of an organism. Whenever I ask you for a genotype, I want like the letters that you're using to code for something, like a pea plant has big P, or a person has big A for they're not going to be albinos. Whatever. So the letters are basically what I want there. If you are just feeling like an overachiever, you could also write out the whole name. Like if a person is big A, that means they're homozygous dominant, and that also can work for a genotype, but it's just easier to just say big A. The phenotype is the outward appearance of an organism. That's also known as the morphology, and you might hear me throw that word morphology around sometimes. But that's just a descriptive for what the genes actually make the individual organism look like. Like, do they have a specific disease? Like in the case of albinism, an albino is a phenotype. In the case of fur color, mice can have white fur or brown fur. In the case of flowers, they can have purple flowers or white flowers. But anytime you're using a descriptor like a color, purple or white or brown, that's going to be the phenotype of an organism. And so you're always going to use words to describe the phenotype and essentially letters to describe the genotype. Next. So usually in high school, they teach you a bunch of really bad science about eye color, and they really should not do that. But I'm going to use that as an example since it's what you have in your head right now, usually from high school. So if eye color is determined by one gene, which it is not, that means based on what they told you in high school, the different alleles for that gene means you can make brown eyes or blue eyes, and apparently high school doesn't understand green eyes or hazel eyes or lilac eyes at all, but those do exist as well. Alleles are those different versions of that gene for eye color, and so pea flowers work the same way. Flowers can be purple or white, so purple is an allele for the gene that is flower color, and white is an allele for the gene that is flower color. In terms of alleles, there can be dominant or recessive alleles as we start talking about this very simply. The dominant allele can essentially hide or smush down the recessive allele so you don't end up seeing it. So a person can have two copies of a dominant allele and look exactly the same as a person who has only one copy of a dominant allele because the dominant always shows up in the same intensity. Recessive alleles, on the other hand, can hide behind dominant alleles, and so unless a person has two copies of a recessive allele, you might not know that they carry that recessive allele. The other note that I have on this slide that's going to be pertinent as we start running through problems, dominant alleles are abbreviated with capital or uppercase letters like capital A or capital P. Recessive alleles are abbreviated with lowercase letters, and so lowercase a, lowercase b, whatever letters we're using at any given time. So as you look at the little picture that's off over here, this chromosome has one allele carried on it, and it's a capital letter, so you know they're carrying the dominant version of whatever this gene happens to code for. This chromosome, on the other hand, has a lowercase letter, so that's telling you it carries the recessive allele. If these two chromosomes were in one individual, it would mean they have one dominant and one recessive allele. Since the dominant is the one that always shows up, or it can beat down the recessive allele, that means they would look whatever capital A happened to code for. Your lab manual starts to have some questions in it at this point. So we're going to come and talk about those flower colors in pea plants again. And remember, you can have purple or white flowers. They explicitly tell you that purple flowers is the dominant allele, white flowers is the recessive allele. What letter should we use for the purple flowered genotype? Let me make sure I turn on, and I'm going to use purple since we're talking about purple. Remember that anytime you're doing a dominant allele, you want to use the uppercase letter, and so big P is a good little abbreviator for purple flowers. So we've already said that should be an uppercase letter. What letter should we use for white flowers? Well, that's recessive. You do not want to use a completely different letter. You just want to use the lowercase of the letter you just did. And so white flowers are going to get a little p. I always write my little p's with this little tail on it when I'm doing genetics problems. That's how I know that's a lowercase p. I really recommend that as you're picking your code for a genetics problem, you always make sure your uppercase and lowercase letters will look very different from each other. Just writing like one big and one small can get really confusing as you start to look up in a Punnett Square what things might have been doing. And so just make your letters look different. They have to be the same letter, but one should be uppercase, one lowercase. So white flowers should be lowercase. Should it be one letter or two letters? So this is where we're going to come back up here to this question. They had asked us for the genotype, and the genotype is the actual genetic makeup of the plant. Since plants have two copies of each allele, they need to have a second letter that's in there. Now, anytime Mendel did things, he did things with true breeding plants, which means we really should have two uppercase P's to answer that question. So anytime we ask for a genotype, there really should be two letters there. Next in the lab manual, they start talking about homozygous versus heterozygous. Homo means same, so homozygous means same zygotes. It means all the babies end up looking exactly the same. In terms of the alleles, that means the alleles are going to be the same. So we're going to either be big A, or little a. We're going to have the same case of letters when we have the genotype for a homozygote. A heterozygous individual is going to have two different letters though, like big A, big B, little b, whatevs. So let's talk about this weird peacock that's going on off over here. First, I want to talk about this side. If you went through high school, probably you heard the term albino get thrown around at some point. Lots of animals exhibit albinism. I've seen albino alligators and moose and, well, peacock and humans, for that matter, can be albino as well. If an individual is an albino, that happens to be a recessive condition. So that would mean we would know that this peacock carried little a because it's the recessive condition. So these cells on this side, they're little a, which means they're homozygous. Now here's where things get really complicated, and it's what high school doesn't tell you, and what honestly the lab manual doesn't even cover at this point. There are complicating factors when it comes into genetics at any point. Mendel got so lucky as he did his results because he happened to look at 7 genes. The majority of them were carried on separate chromosomes. He got the numbers he got because of that. Reality is, again, much, much messier. So here we have a peacock that's half albino, but half normal. This is what's known as mosaicism, and it's the reason why a person can have one blue eye and one brown eye. It's because that person is a mosaic. They have different cells that have different combinations of those alleles. These cells off over here, and you can't see the purple on the peacock, you know they have to have at least one capital letter because they are exhibiting the colors that a peacock is supposed to have. You don't really know what his second letter is. Those cells might be big A, or they might be big A, little a. They're probably big A, little a, but we actually can't know for certain. That means if that peacock were to have sex and make some babies, we don't really know what kind of babies they would make because we don't know if these are the cells that might make this is a male peacock, so he would make sperm. So we don't know if his sperm came from that kind of cell, or does he make sperm with this kind of cell. So this is just one of those weird, complicating factors that I always found fascinating when it came to genetics, but it does make things really complicated in reality in a lot of cases. Okay, I already kind of alluded to this fact earlier. Anytime you see the word true breeding, it really means inbred, and plants can inbreed to an extent that even humans can't. Like, you can breed a plant to itself. What it really means is anytime you see true breeding in a problem, it means they're going to be homozygous, and so they're going to be big P, or they're going to be little p, little p. So the next question that it asks you It's talking about in Mendel's crosses, he would always start with true breeding plants, and he bred purple flowered plants together with white flowered plants. So what's the genotype of the purple flowered plants that he started with? Well, since they're true breeding, they're homozygous, and so they would be big P, big P. Next, it goes into the generations. Anytime Mendel did an experiment, he would always breed through 3 generations. The ones that he started with were the parents, and so he called them the P generation. The kids of the parental generation is the F1, so in the picture, he bred these 2 together. Anytime you see that X, it basically means they're doing it, although plants doing it is not as fascinating as other organisms doing it. But anyway, those 2 had sex, they made babies. Their babies are the F1 generation. Then, because plants can do this, they would allow this plant to self-fertilize, which means it would have sex with itself, but in a planty, polleny way. And then they would make babies, which were the grandkids of the original P generation, the kids of the F1 generation. So we've got parents, kids, grandkids. That's how the generations work. He never extended past that in any of his writings. The Punnett Square is the box that we're going to use to help solve problems. It's usually going to be a 2x2 box like you see in this picture. In lecture, we're going to do some dihybrid problems, which means we'll get 4x4 boxes, but we won't do that in the lab. At that point, the lab manual has a bunch of other definitions, like they define true breeding again, and self-fertilize versus cross-fertilize. Make sure you read through those so that you understand those definitions, but I'm not going to write them down for you again in a PowerPoint. So, at that point, we have our first example problem. It goes back to the flower colors that Mendel was working with. So he bred his true breeding purple flowered plants with his white flowered plants, and all of the kids ended up being purple. That helps us answer the next questions. Which phenotype is dominant? It's got to be purple, because remember that white's there, but it's hiding. That means the white is recessive, and purple must be dominant. Since purple is dominant, what letter are we going to use for that? Big P. What letter are we going to use for white, since it's recessive? Little p. What is the genotype of the purple parent? Because it's true breeding, it must be big P, big P. What is the genotype of the white parent? Little p, little p. We know that for 2 reasons. 1, it's true breeding, and again, all true breeding plants are going to be homozygous. But 2, it has white flowers, and the only way you can have an organism show the recessive allele is to have 2 copies of the recessive allele. So since it's white, it must be little p for white, white. Next, fill out the Punnett Square for that cross. Okay, so we've got our purple flowered plant and our white flowered plant. In order for us to fill in the square, we've got to know the gametes. The gametes have half the letters in them as the original parent, which means this parent can produce a gamete that is big P, or it can produce a gamete that is the other P, so still big P. This parent, and I'm going to change colors in the hopes that it will help you see kind of how things are getting passed down. Oh, I always forget I have to escape. One of these days I'm going to remember to do everything I'm supposed to do. Y'all will see. I'll do it. Okay, so next. The other parent we're going to put on the side. So little p from one of those little p's, and then the other little p will go right here. After you have put the gametes across the top and down the side, the next thing that you do is you bring the letters that are in the top down into every box that is below that. So I'm going to switch back to the original color that I use. This big P is going to go down here and here. And then this big P will go here and here. Then the letters that are on the sides, you take them into every box that is beside them. So this little p is going to go here and here, and then this one will go here and here. So, next question that it asks you is, what is the genotype in each of the boxes? Well, as you look at that, they are all big P, little p. So they are heterozygous. Remember, if I ask you for a genotype, you can just provide me the letters like I wrote here, big P, little p. Or if you wanted to be that overachiever, you could say they are heterozygous, which I just answered the next question. They are heterozygous. What is the phenotype in each of the boxes? So this one's asking you, what do those look like? They have a big P and a little p. They're going to look purple because the purple is the dominant allele, and so that's the one that we're going to see. What generation are in the boxes? In the boxes, so we put the parents outside on the top and on the side. That means when we bred them together in the box, inside the box, we have the F1 generation. Alright, next. He would allow one of the F1 offspring to self-fertilize, which again means having sex with itself. So what cross did he make? Big P, little p, big P, little p. We're going to take those, write one of the parents on top, and then I'll still switch colors just in case it helps any of you out there. The other parent goes down the side. Fill it in, same as always. Now, one of the things that students usually ask me is, does it matter where you write the letters in the box? And it doesn't. So I just brought the side stuff over. Now I'm going to bring the top stuff down. Notice again I'm color coding things. Okay. Now, here's what I mean by the students will ask me, does it matter where they write the letters? Here, just for ease, I brought that little p over and put it towards the front of the box, and so this makes it look little p, big P, versus this is big P, little p. Those are still both the exact same thing. They're still both heterozygotes. And so it really doesn't matter if you write the uppercase letter first or second. Remember, these are chromosomes inside of a cell. It's going to get messy inside of there. It's really just about getting the right letters in the right box. It doesn't matter where you put them in that box. So, what are the genotypes for each of the boxes? So, in this first box right here, we have a homozygous dominant, big P, big P. In those two boxes that I was just talking about, we have two that are big P, little p, or heterozygotes. And then we got that poor guy, little p, or homozygous recessive. So, what is the genotypic ratio? This was one of those other definitions that I mentioned earlier. If we ask for a ratio, we want a number to a number, possibly to another number. And you have to tell us what those numbers actually mean. So here's how we would write that. We would say there's one big P, for every two big P, for every one little p, You would not just say 1 to 2 to 1, because that doesn't tell me 1 of what to 2 of what. For all I know, you solved the problem wrong. And so you have to make sure you're telling me what the numbers actually mean. What are the phenotypes for each of the boxes? Now here's where I'm going to switch colors again, since we're not talking about genotype anymore, we're talking about phenotype. Remember that if there's even one big P, that plant is going to end up with purple flowers. So all three of these end up with purple flowers. So we've got 3 with purple. They didn't ask us for numbers just yet. And then that last one that I didn't circle is white. Once again, I'll apologize for my messiness. It's really hard to write on the screen for me. What is the phenotypic ratio? Same thing, we want numbers, and you have to tell me what the numbers mean. So now we're going to say we have 3 purple for every 1 white. What generation are in the boxes? Well, this time we had the F1 here and here, which means if you cross the F1 together, you get the F2. So the F2 is in the boxes. Next, your lab manual starts to talk about test crosses, and they first ask you some questions, and then they kind of tell you what a test cross is, and I wanted to tell you first what a test cross is. It is possible for you to have an organism that is showing the dominant phenotype, like a purple flowered plant, but it's possible for it to still carry that white allele that's hiding behind the purple. In other words, it can be either homozygous dominant or it can be heterozygous. A test cross finds out for sure what your organism is. Is it homozygous or is it heterozygous, barring a lethal mutation or something. So, what you would do is you would cross your unknown dominant organism to a complete recessive, and then you would look to see what the ratio of the offspring were, and depending on that ratio, you would know whether your organism was going to be homozygous or heterozygous. So from that description, we're now going to go ahead and run through and answer these questions. So it says, pea plants, in addition to the flower color stuff we were working with, they can also have different versions of their seeds. The seeds can be yellow or the seeds can be green. It says to cross a yellow-seeded plant with a green-seeded plant. What uppercase or lowercase letters would you use for yellow? Well, since it says that's dominant, we do a big Y for yellow. What uppercase or lowercase letter would you use for green? Lowercase y. And again, I give my lowercase y a tail so that I know that's the lowercase. Mendel is crossing to a green-seeded plant, so what is the two-letter genotype of the green-seeded plant? Well, since green is recessive, and we abbreviated it with that lowercase letter, in order for a recessive plant to exist, it must have 2 copies of that recessive allele. So it would have to be little y, little y. Mendel is crossing a yellow-seeded plant. What is the two-letter genotype of a yellow-seeded plant? And they even say it's a trick question. Well, that's because, remember, there's 2 options for a plant to be yellow. It could be homozygous, big Y, or it could be heterozygous, big Y, little y, and still end up being yellow. So, we just kind of had this problem a second ago. We had the question of what is the yellow's genotype. So I already said that stuff up there towards the top. They want us to go ahead and fill out these 2 Punnett squares. So we've got this one with a homozygous dominant being bred against a green flowered plant, and this one, heterozygous yellow, being bred against a green plant. So once again, bring the top stuff down, side stuff over. So it looks like that. Okay. Which Punnett square, A or B, has offspring with all yellow seeds? Well, as you look at this one, every one of those boxes has at least one capital Y, which means every one of those babies end up having yellow seeds. And so the correct answer to that question is A. Now I want to go ahead and look at B really fast. As we look at that, these 2 are both yellow, these 2 are both green, which means this one gives us a 50-50 split. So earlier I said we have a plant, in this case, that has a dominant phenotype. It looks yellow, but we didn't know if it was big Y yellow or big Y little y yellow. By breeding it against the known recessive, if 100% of the babies are yellow, that told us that our original parent must have been homozygous. But if in the process of doing that cross we end up with 50% yellow peas and 50% green peas, well, that tells us that our original yellow must have been heterozygous and not homozygous. That's the value of a test cross. It tells us what the dominant looking plant is from a genotypic standpoint. Is it homozygous or heterozygous? All right, so continuing on with that test crossy thing, which Punnett square had offspring with half yellow seeds and half green seeds? As we go back, that was B. So B. Now, what is the genotype of the parental yellow-seeded plant? Well, in B, the genotype was big Y, little y. They were the heterozygote. Okay. From there, they start to tell you that sometimes in a genetics problem, you have to kind of work your way around backwards. So we are back to working with pea plants, only now we're looking at how tall the plant is. They could be tall, which is dominant, or short, which is recessive. He crossed 2 tall pea plants together, and then he ended up with 75 tall plants and 25 short plants. So what kind of a cross did he make, is what it's asking you. So now is where we're going to start working our way backwards and just answering the questions they ask you. So what letter should we use for the tall phenotype? Since it's dominant, capital T. What letter should we use for short? Lowercase t. And again, my lowercase t's get a little tail on. What is the 2-letter genotype of a short plant? Since short is recessive, they must be lowercase t, lowercase t. What is the 2-letter genotype of a tall plant? The tall plant, let me say that differently. So as we come back to this problem, Mendel had ended up with 3 tall plants for every 1 short plant. That does tell us something. And like she says in the problem when she wrote the book, some of you are going to get right away that you know exactly what the genotype must have been for that tall pea plant. Some of you don't know, though, yet where that is. So we're going to do that whole work our way backwards sort of a thing. If some of our baby pea plants must have been short, because remember 25 out of the 100 ended up being short, then we know that within our Punnett square we must have a big T, or pardon me, a little t situation going on. In order for this box to be filled in like this, that means up here there must be a little t, and here there must be a little t. Now, going back to the original problem, it says He crossed two tall plants together. In order for these parents to be tall, that means their other T must have been a big T. So, coming back, what is the two-letter genotype of our tall pea plant? Must have been heterozygous. Big T, little t. So, where did each lowercase t come from for that 25% of the plants that ended up being short? It came from this recessive allele that was hiding behind the dominant allele in both of those two parents. Okay. After that, we start talking about probability, and probability is all about math, which I learned a long time ago. Texas students, the second you say math or start doing addition or multiplication, eyes start to glaze over and fear starts to come out. We're going to keep this really simple as we start to talk about things. And you can use a calculator if you really need to. So we're going back to the purple versus white flowers. We're going to self-fertilize a big P, little p plant. What is the probability of getting a purple flower? What cross are we making? So we are doing big P, little p times itself, again, inbreeding. Fill in your Punnett Square. So big P, little p, big P, little p, top stuff down, side stuff over. Fill in your phenotypes. These three are purple. This one ends up being white. That's me filling in my phenotypes. How many purple flowers are possible? Out of that box, we have 3 purples for every 1 white. So how many purple flowers are possible? 3. How many total boxes are there? 4. So the probability of getting a purple flower, you actually just make that a fraction. 3 out of 4. Now here's where I said you could use your calculator if you really wanted to. I'm perfectly happy if you tell me 3 out of 4 chance for a probability, but sometimes on the problems I will give you a percentage instead of a fraction like that. And if I ask you that, you could literally do 3/4 on a calculator by just doing 3 divided by 4 and then make that a percentage. But that would be a 75% chance of producing a purple flowered plant. So, what is the probability of getting a heterozygous genotype from the same cross? Well, this time it's only asking about heterozygous. That's just these 2 out of 4. So it's the number of heterozygotes divided by 4 boxes. So 2 out of 4. You can, reduce that to 1/2. Or again, you can write the percentage. It's 50% chance that you get a heterozygote out of that. Now here's also where I really need to hit something home that's really important for you. I am not saying that when you allow this plant to self-fertilize, it's going to have exactly 4 kids, and these are going to be the 4 kids that they end up producing. No. What I'm saying is, these are the possibilities that they can make if you allow this plant to self-fertilize. While it would be unlikely, it's entirely possible that you breed that plant together and you get 2 babies, and they both happen to be white. That's a bias sort of a situation, but it's still statistically possible. It's just not probable. And so you need to understand that anytime we do these problems, all we can ever tell a person is what's the probability that their kid ends up looking a certain way. After that, they start to talk about, well, what's the probability that they have a purple flowered plant and then a white flowered plant? We're not going to get into that, and so you can just skip that whole section. If you really want to play around with it, it's called the multiplicative probability, and you have to multiply the probability of each of those individual events times each other, and it will give you a smaller chance that both of those 2 events happen and in that order. After that, a bunch of questions for us to answer. So, dominant allele is always expressed. The box diagram is the Punnett Square. The recessive allele can be hidden. The different version of a gene is an allele. The genetic makeup of a cell is the phenotype. And the visible trait is the phenotype. And so that's the order that those numbers should go into that problem. Next, for each genotype, indicate whether the genotype is homozygous or heterozygous. Remember, if it's 2 of the same letters, homozygous. 2 different letters, heterozygous. And so, same homozygous, different heterozygous, same, different, different, same. For each of the genotypes below, determine the phenotypes. Green seed pods are dominant to yellow seed pods, so that means capital G is going to be green. So if there's at least one capital G, it's going to be green. So that's green. This is green. This one doesn't have green, so the opposite of green here is yellow. Smooth seed pods are dominant to wrinkled seed pods, so capital S is going to mean smooth. Lowercase s is going to be wrinkled. Wow, that got extra messy towards the end there. Hopefully you write neater than me. Okay. For each phenotype, list the genotypes. Remember to use the first letter of the dominant trait as you make your codes. Purple flower is dominant to white, so homozygous dominant, big P, big P. Heterozygous, big P, little p. Homozygous recessive, little p, little p. Yellow is dominant to green, so big Y, big Y, big Y. Little y is heterozygous, little y is homozygous recessive. In pea plants, tall is dominant to short. Cross a homozygous tall plant, homozygous tall, two big T's, to a homozygous recessive, lowercase t, lowercase t. So that's the genotypes of each parent. Which gametes can each parent produce? First one can make either big T's. Second parent can make little t's. Fill in the Punnett Square. Big T, little t, little t. And then bring stuff down and over. What are the phenotypes of the offspring? Remember, phenotype is what do they look like? They are all tall. What percentage of the offspring will be tall? 100% of those are tall. And pea plants, tall is dominant to short. Self-fertilize a heterozygous pea plant. Heterozygous means two different letters. And if we're self-fertilizing, it's both the mom and the dad. What gametes can it produce? For this parent, big T, little t, or big T, little t. We'll fill those in around the Punnett Square. And then top stuff down, side stuff over. What are the genotypes of the offspring? Genotypes means our letters. So we've got big T, big T, little t, or little t, little t. What percentage of the offspring will be heterozygous? These are the heterozygotes, so 50%. Tall is crossed with short. What percentage will be tall? Again, there comes a point where you just look at a problem and you know the answer, but for those that don't, if we're crossing tall with short, every single box is going to look exactly like that. And I gave that a tail, I didn't really mean to. So, 100%. Heterozygous round-seeded plant is crossed with a homozygous round-seeded plant. What percentage are going to be homozygous? Top down, side over. And it only asks, this time it's not asking how many are going to be round, it's asking how many are going to be homozygous round. These are the homozygous round, so that's 2 out of 4, or 50%. Yellow seed color is dominant to green. We're going to test cross a yellow plant. Remember, anytime you see test cross, that means one of your parents automatically is going to be recessive. The F1 generation has all yellow seeds. What is the genotype of the yellow seeds? Okay, we did this, I think, exact problem earlier. Maybe we didn't do it with the seed color, but we know that one of our plants had to be the known recessive if 100%, where did it say that? Hold on. There we go. The F1 generation all has yellow seeds. Well, since our known recessive only gave green seeds to these, that means the yellow must have come from the other parent in both cases. So if you're backwards working, that's how you backwards solve that problem. The other thing is you can just remember if 100% of the babies all look yellow, that means your parent plant must have been homozygous, and so it has to be big Y, big Y. What is the genotype of the F1 plants with yellow seeds? So that's asking you what's going on in the box, so they are all heterozygous. Seed color again, we're test crossing again. This time though, half are yellow, half are green. So we'll do it this way. Remember, one of our parents has to be little y, little y. Okay. We know that we need half of them to end up being yellow, but the other half can end up being green. And so this time we must have had a heterozygote. Plant seed colors one more time. This time the generation has about, oh, this time it doesn't say test cross. I'm sorry, it just says crossed. The F1 generation has about 3/4 yellow seeds and 1/4 green seeds. What are the genotypes of our parent plants? Okay, for this one, in order for us to get both yellow and green, but to end up or to start with plants that have yellow seeds, that means we're going to have to do heterozygotes to start with. Then as you fill in your squares, you will see that 3/4 of them, those three that I've already written down, end up being yellow, and 1/4 end up being green. So that means the genotypes of both parents must be big Y, little y. Seed color, this time we're crossing green plants. Remember, if they're green, that's recessive, so all of our parents are little y, little y. And if everything there is all little y's, that means all the babies got to be all little y's. I'm not even going to finish the cross because it's just homozygous recessive, little y, little y. What color are they going to be, though, is the question they ask us. And these guys are all going to be green. Don't get weird on me. Green. One of the other mistakes that students very often make is they don't actually answer the question. They'll think they know what question was going to be asked. Like, I thought they were going to ask what's the genotype of the F1 generation, but they didn't. They asked for what the color is. So just when you're solving a genetics problem, make sure you go back and read the question so that you know exactly what they're asking you for. All right, I think this is the last one. NP plants, tall, dominant to short. He crossed a tall plant with a short plant. He counted 80 tall for every 20 short. That ratio is closest to, out of the options that they have given us, it's closest to that 3-to-1 ratio. So what cross did he make? Well, anytime Mendel got a 3-to-1 ratio, it was because he was crossing two heterozygotes, a big T, little t situation, like this. As we fill it in, what you will see is the same thing we've seen over and over again. You end up with 3 that have the dominant phenotype, tall in this case, for every 1 that has the recessive phenotype, which is short in this case. So he crossed 2 heterozygotes, big T, little t times big T, little t. Make sure that's the last problem. There we go. So make sure you complete the post-lab quiz over genetics. If you have any questions, please email